Anyway, I'm coming here today from the perspective of a Water Policy Advisory Council in trying to deal with an issue that keeps coming up for us, and I'm sure it comes up for other WPACs and lots of other people as well. And that is, what are all these weird organic chemicals that people keep reporting in water, and why are they important? Is it a health issue? Is it any other kind of issue? Our problem, of course, is the same as others, and that is that we don't have a lot of information, and the data that we do have are limited, but they're quite expensive to come by. So our challenge then is, how do we work with a very limited database and try and interpret this information for the public and make it intelligible to them and hopefully reassure them that the sky is not falling. Just to say a word or two about the other members of the team here, because this is very much a local effort from our WPAC and Medicine Hat. I took the lead on this. Grayson Mouch is actually the Environmental Utilities Director for the City of Calgary. And Ryan Davison is a PFRA guy who just got downsized, so he's got a little time on his hands, and he was happy to help out. Morgan Baziak and Rachel Brown are both students. And Morgan was, in fact, the one who I'd hoped to have here today, because she could have presented this paper just as well as me. But she's a third-year pharmacy student at the University of Alberta and couldn't get away today. But she's the main brain there in the lab, and also about pharmaceutical compounds of all descriptions. In fact, she's probably forgotten more than I ever knew about them, but whatever. And Rachel Brown was actually a graduating grade 12 student who worked in the lab last summer and assisted Morgan and did much of the lab work. So we are essentially the local team. So what it came down to for us was that we were getting lots of questions that we had no answers to. And the questions centered around, what are these compounds that get into water, things like pharmaceuticals, pesticides, herbicides, industrial chemicals that run off from the environment, microsystems from algae, many other things. Of course, there are lots of possibilities. Very few people who ask about water quality really have a good understanding about what they're asking. They don't really understand what water quality is, and we've had this challenge before that even with conventional water quality data, there's really no use in giving people a whole lot of spreadsheets because they can't interpret them. They're no use to them. We need to be able to take these data in, we need to be able to assimilate them in some way and present them in a manner that's intelligible to the average person so that we can give them some assurance that the water is or is not in good condition. So we launched this project as a pilot project, essentially. Our problem, again, was that we had few data and we had no resources to go out and generate many more data because these samples are extremely expensive to analyze. You could easily spend $2,000 to $3,000 on one sample. So clearly we're not going to get a big database from that. But as a pilot project, we wanted to evaluate new methodology that was originally developed by the U.S. Department of Agriculture and is currently being marketed through a company called Abraxas in the United States. Now, I have to tell you right now, this talk is in no way sponsored by Abraxas. We bought their kits, that was it. But the method that they employ is based upon pretty conventional biochemistry. So instead of using a gas chromatograph and mass spectrometer approach to analyze samples, we're using ELISA-based technology, which means that it involves antibodies and enzymes and pretty conventional stuff. The big breakthrough that the USDA came up with on this was to be able to produce antibodies against these compounds that are of interest for various reasons. There's quite a wide variety of these kits available. They come either in two formats. They're either in an ELISA plate, which is a teeter plate containing 96 wells, so you get 96 tests in a kit, or they come as an immunogenic bead kit, where the bead itself is used to attach to the target, and it is extracted from the sample using magnets. So these are both conventional things that are used in laboratories. I could explain more later, but you don't want to hear about all that right now. If anyone's interested, I'll be glad to afterwards. But there's no reason why this shouldn't work, basically. But we just had to try this out and find out if we could actually get any useful data from this. So we launched it last summer as a pilot project with the aim of showing whether or not it could work, to try and validate the approach. We were not trying to monitor or collect a large amount of data. We simply didn't have the time or resources, and it wasn't the time to do that either. We needed to make sure that this was worth pursuing. So just a bit of background. Organic material is common in surface and groundwaters, and much of it is natural. It consists of humic and fulvic acids, tannins and lignins, small amounts of carbohydrates, proteins, and so on. There are many things that might be there. And there's no particular reason to believe that there's any problem in the South Saskatchewan River here. These are fairly normal values that vary seasonally. So we didn't go into this expecting, or we didn't know what to expect when we went in, whether there would be any big issue with these compounds or not. But just as background, dissolved organic compounds are there. They're usually in quantities of several milligrams per liter. But the things that we're interested in are only present or likely to be present at much lower concentration, in micrograms per liter or in nanograms per liter. So here we are down here at the bottom of the province, and we're downstream of just about everybody down here. So we always assume that whatever comes from up here is going to wind up down there, and this is not good for us. There are several problems with this whole general area, and the first one is that there are literally hundreds of compounds out there, all sorts of toxic things, or things that might be toxic. There are any number of pharmaceutical compounds. There are other ones that are probably harmless, but are there and sometimes as useful markers, things like caffeine, for example, common in effluent, and will wind up in our surface waters. The analysis of these compounds is difficult and expensive by conventional methodology, and that's not likely to change, but very much a limitation on how many data we are likely to ever accumulate. We also don't know very much about the harmful effects, if any, on many of these compounds. Now, some of them, if we're talking about DDT or something like that, we can relate to that. We know a fair bit about compounds like that, but many others, we have no clue. There are no guidelines for most of them. Very little research done on them, and the concentrations that have been reported in the environment are often quite low, and so it's difficult to know whether they really are having any effect or not. Our approach then was, first of all, to gather as much data as we could, because we did need to answer this question that we keep getting asked, and that is, what is the importance and significance of these compounds in water? And the approach we chose was to choose representative compounds. Now, that's the same problem we have in microbiology, and that I can speak more authoritatively about because that's my background. But in microbiology and water, when people want to test for that, you can't possibly test for all the organisms that could be present in water that might be pathogenic. There are far too many of them. It would take too long, it would be too expensive, and by the time you got the answer, nobody would care. So we choose indicator organisms. We use total coliforms and fecal coliforms and other organisms like that, and we make the assumption that if they are present, then other more serious pathogens could also be there. So why not use the same approach for trace organic compounds? If we're looking at banks of literally hundreds of them, even thousands that could potentially be there, why not choose some representative ones, monitor those, and if the methodology seems to be satisfactory, we could generate some useful data, hopefully, that would at least give us an indication of whether or not we were looking at a problem. So we used this methodology that was developed by USDA. If anybody's interested, you can check it out on the web at abraxaskits.com. And we also recognize the need to be able to validate any data that we produce with more conventional methodology. Nobody's going to believe numbers that we produce if we can't back them up with more classical methodology and show that we're getting reasonably comparable results. And so to do that, what we did was using the City of Medicine Hat's resources. They occasionally need to do these kinds of analyses on drinking water and do it even sometimes on raw water, just for their own records and to make sure that they're in compliance. So using those data and analyzing the same samples with the kits that we had and the methodology we were using, we hope to validate the data that we were receiving. A little bit more background here, just to give you some idea. Even though we are downstream of just about everybody in the southern part of Alberta, our water quality really isn't that bad. You know, if you look at the water quality that we have here, here's us, we're generally in the pretty good range here. This is a surprise to many people who live in Medicine Hat, who always assume the worst. So we also then moved on to see what the existing information database was. And there's some out there, there's not a lot. As someone mentioned earlier today, the information that is available tends to be rather scattered and hard to come by. But we pulled it together as best we could, and we probably missed things, so if anybody knows of something that I did miss, please let me know. The first study here was one on pesticides, and here we see a number of different river systems here. Here's us in the South Saskatchewan River. Looks like the number of exceedances here was actually, or the detection frequency was pretty low for us. Much higher in the Old Man, the Red Deer, and the Bow, and North Saskatchewan. And this actually was the beginning of a bit of a theme that was emerging from our data from here and from microbiological data that we looked at in other contexts, was that when we're talking about contamination in Alberta's river systems, it's largely a headwater story. We keep expecting to see more and more stuff down at our end of the thing, at the very bottom of the river system, but it's not there in many cases. But I'll return to that later. Here we're looking at the pesticide measurements that actually exceed the surface water quality guidelines such as they exist in different categories: drinking water, irrigation, livestock, and freshwater aquatic life. And in this study, we find again that the South Saskatchewan River is not showing up nearly as often as the Bow or the Oldman River, which are, of course, the two major river systems that make up the South Saskatchewan in our part of the province. And here are a number of different compounds that might conceivably be of issue here. Common herbicides. Glyphosate is the Roundup one. That's another issue with these things, by the way. You get so many big names, and many of them don't mean anything to anybody, that it's hard to know whether it's really a problem or not, but the names themselves look scary. And here we have some pesticides and other things there. So what we're seeing in our part of the world down here is dicamba, a common herbicide, and some mecaprop, which is another common herbicide, which is not a surprise. If you look at where we are here, this is where Medicine Hat is down here. Here is the Bow, and here is the Oldman that make it up. This is a very large agricultural zone here, heavily irrigated through the St. Mary's Irrigation District. If there was to be any return flow there, you might expect that there might be herbicides and other things there. But there really are not very many other sources that you can put your finger on that would be large, except for the city of Medicine Hat. And the other interesting thing about this is that Medicine Hat is here, and there is only grazing land from here down to the border. So there's very little chance for other effluents other than potentially agricultural ones to get in there. There are no towns dumping effluent in and things like that. So it makes for an interesting situation that is certainly going to be something we'd like to pursue. Another large study that took place back in 2005 was just a preliminary survey that was done by Al Sosiak and Theo Heaven. And this looked at pharmaceutical compounds and endocrine disrupting compounds in both wastewaters and the receiving waters in Alberta. So this was a broad spectrum kind of survey. It was based on single samples taken at most sites, and so it was not pretending to be a comprehensive monitoring exercise at all. It was merely a study to see what might be out there. So there were a lot of data in that report. You can download it from Alberta Environment's websites, available there. But the data are too numerous to report in detail, but I'll give you some idea here. This isn't going to be very visible in the back, but the main point is that, first of all, we have effluent here, that is sewage effluent, and over here we have river concentrations. So these three graphs are rivers, and these three are the ones from effluent itself, where you would expect to find many of these compounds. There are a number of different river systems identified here. Medicine Hat water treatment in the South Saskatchewan River, that is in the yellow on this side here. So again, we're pretty low on most things, but not always. Here's effluent, but down in the river, now we're green, just to be helpful, but you can see we're pretty low. Not a whole lot going on there. But again, it was only based upon one single set of samples taken on one occasion, and you really can't say much more than it's there, and it doesn't seem to be a lot of it, and it's common. You really couldn't conclude too much more from those data. These EDCs are endocrine disrupting compounds. They are hormones. They're going to be things like testosterone or estrogen or anything that mimics those compounds, and they might conceivably have a bad effect of some sort on aquatic life, particularly fish. You know, we wind up with gender-confused fish, and most people have seen episodes of The Simpsons where you see all these horrible things down there, and that's about the limit of their knowledge on that, and it's pretty scary, so we need to know more about this. Anyway, it's basically good news down here for steroid estrogens. In our part of the world, there are hardly any. These are actually surrogate-type compounds that are not even the straight hormone themselves. So, not too much to get excited about there, but we know it's out there, certainly in effluent. Here are some data from another study that was done with pesticides from the heavily irrigated zone that's in our watershed. This little inset map here shows parts of the St. Mary's irrigation river system. Here's the South Sask, the Bow, and the Old Man. And these samples were taken in a number of different locations, either from return flow, which was R4, that's the circle, and would show up, the solid circles are lost in here somewhere. And they would be open channels, which is what you tend to see from the highway, those great open channels that are flowing all over the place. So that would be these guys here, E1, 2, and 3. And then there were some pipeline sites as well, because water's piped at different places in the St. Mary's system to get from one place to another. Also, I have to admire their audacity in just drawing those lines straight across there for a whole year, but you can see that they only took samples here during the warm season when the canals are actually flowing. You don't get much samples out of the irrigation system in the middle of winter because there's no water in them. But again, the concentrations are pretty low. This is 2,4-D, the common ingredient in compounds like Killex and whatnot that we can't even buy in Medicine Hat anymore because they've been basically outlawed. And the city no longer sprays these too, which is interesting. So in those days, at least, this is showing it's there, but really this is micrograms per liter, less than 1 microgram per liter, and these are, in some cases, irrigation return waters. If we go back here and look at these concentrations here, these are actually in nanograms per liter. I mean, we're talking very small units here. So even though the peaks may look large, the scale is with very low concentration. So. And lastly, a bit more about what we found. Natalie Cromery did her master's degree thesis at the University of Lethbridge looking for effects of sewage effluent on the reproductive capacity of a common minnow, the fathead minnow. And she was able to find some evidence that the effluent itself, not the river water, but the effluent, was able to at least stimulate some liver function changes here because of the exposure to estrogen mimics. And she was unable to conclude much downstream because she had a hard time finding a large enough supply of fish, but there wasn't a whole lot of activity. And there seemed to be no significant effects at all from, at least as measured by this enzyme, acetylcholinesterase, from pesticides. And that's rather a surprise, because if there's anything you'd expect to have lots of down in our end of the river, it would be herbicides and pesticides from agricultural runoff. It would also be logical to look at what the city's putting into the river, but a bit of math there is illuminating too, because the output of the sewage treatment plant could not be more than about 30 megaliters a day, because that's all it can handle. And that is about what you see throughout the year, because it tends to be fairly constant. That constitutes less than 1% of the discharge in the South Saskatchewan River at the effluent point, even at low flow. At high flow, which could be an order of magnitude higher, it's correspondingly less. So the amount of effluent that's actually getting into the river is small relative to the actual volume flowing it. This would be less true upstream, and I return to that theme that I mentioned earlier, that if we're looking at systems like the Bow or the Old Man or even the Red Deer, then you're looking at smaller discharge because the rivers are smaller at that point and more people. So the effluent treatment at those sites becomes increasingly important because it's going to be potentially a larger, not necessarily a dangerous, but a larger proportion of the actual flow in the river. So some evidence from fish, not much. That's all we have there. So here's what we looked at. We looked at all the compounds that we could think of and all the major classes that might likely be in effluent or in discharge from agricultural areas, and we chose a dozen or so that seemed logical to us that would be worth monitoring. We tried to represent major classes of compounds with easily recognizable and measurable compounds with these kits. So we wound up with 2,4-D, logical choice for a pesticide. Actually, an herbicide would be more logical to call that, but anyway, 2,4-D. Glyphosate is the one in Roundup. And there's an issue with Roundup too. The surfactant that's used in Roundup can actually be more toxic to aquatic life than the Roundup itself, so you have to be careful what you're measuring with this. But we had no way of doing the surfactant. We had to stick with the Roundup itself, or with glyphosate. There are 3 antibiotics that we chose. Sulfamethoxazole is a common one in humans. It's the one that you probably gave your kids when they had an ear infection that comes in the pink liquid called Septra or something like that. Sulfamethicine is equally common in animals, and so are avermectins, which is an antiparasitic compound that's commonly used in veterinary use for large animals. For antimicrobial compounds, we chose triclosan, which is that common ingredient in all sorts of pharmaceutical products, especially hand soap and detergents, that is antibacterial. There's been a fair amount of controversy about this in the last few years because it is suspected that it may be capable of inducing antibiotic resistance in native bacterial communities in either water or soil systems if they are exposed to it, which would not be a problem for triclosan especially, but it might be a problem if it induced antibiotic resistance in, to more commonly used drugs in medicine. So that was an issue. We also looked at a couple of industrial chemicals, benzopyrene, which is a common byproduct of a number of different industrial things, particularly combustion, but it also has natural sources as well. So it's not entirely a pollutant, at least not a man-made one. And alkyl phenols, which are estrogen mimics in some cases and may also induce some gender confusion, shall we say, in fish. And then the hormones themselves, estrogens. Ethinyl estradiol is the one that's in birth control pills, slightly different from estrogen and testosterone. And then for algal toxins, microcystin, which is, of course, an issue for us down in our corner of the province because there are lots of sloughs and other surface water bodies that contain lots of blue-green algae that could produce this stuff at different times of the year. So we thought, let's monitor those and see what we get. The big problem we had with actually saying whether what we found was important or not is that there are no guidelines for most of these things. These are all that I was able to find in a search in the literature. There may be more out there, and I'm only looking at Canadian ones here. I didn't look at EPA or other countries. We could add those in. But for most of these things, there are few guidelines and therefore few yardsticks to measure their importance by. So what did we find? In the river, we took some samples last summer because last summer we were just getting the methodology up and running, so the number we actually managed to analyze was fairly limited. And at that time, this is in late July and August, there was still a lot of snowmelt in the river, high river water, and therefore more diluted than it would be in the middle of the winter. So we then went out and took another sample that we were able to obtain on the 28th of December, on a lovely day out on the river, at low flow. So this should represent the absolute minimum amount of input, at least from agricultural sources, but it might potentially be a larger time of input from the sewage effluent from the city of Medicine Hat because it's more or less constant at 30 megaliters a day and the river discharge was down. So at that time, we were also able to submit the same sample from the very same bottles to ALS laboratories for their classical GCMS analysis to see what kind of numbers they got compared to what we got. So for this sample here, we do have other data, but this was the best set we had from December the 28th. All of these compounds were in reasonable agreement. We have a couple of issues here. One was benzopyrene. They got less than 5 nanograms per liter, and we got 1,160. Don't know what the story is there. And although it doesn't show it here, we also have a bit of a question about the avermectins analysis too, but unfortunately the ALS system did not analyze for that, so we have nothing to compare it with, but it looks suspiciously high. And yet suspiciously high in this case is less than a microgram per liter, or at least in that neighborhood. This scale here is in nanograms per liter, very low concentration. So even though we got a few peaks here for things like 2,4-D and for microcystin, the algal byproduct, everything else is vanishingly small. And this is what you get back from places like ALS laboratories too. You could get 5 pages of tables of data of things that they analyze for, and almost all of it is below detection limit. So it doesn't tell you whether it was even there or not, it's just below detection limit. And the detection limits are often quite low. So that's not very helpful in terms of showing what sources there might be out there, but at least it's encouraging that whatever it is, it's not a huge problem, not in terms of concentration at least. So those were the validation efforts that we've been able to do to date. The reason we don't have more, by the way, is that this is an unfunded project. It's been scraped together out of volunteer labor by donations from the city of Medicine Hat, who actually gave us the kits themselves, and with a little bit of student stipend funding we had last year from the Alberta Association of Colleges and Technical Institutes. But otherwise, we're on a real shoestring here. Some more data from last summer and from a bit from December the 28th, this one winter sample that we were able to take, showing all the different major classes of compounds here. Not a whole lot going on with the possible exception of avermectins. We need to check this one out and see. It may be that whatever that kit's measuring is not the same as what ALS is measuring. and the same is true of benzopyrene. But other than that, we're not seeing a great deal. This is concentration in nanograms per liter, and the sample came from Ross Creek, which is an important, not a large, but an important tributary to the South Saskatchewan River that actually flows straight on the outskirts of Medicine Hat. It drains a large agricultural area, lots of livestock and other ranching kinds of activities there. Similar data from Seven Persons Creek. Seven Persons Creek also drains lots of agricultural land, and we chose two sample sites for that. One of them is at the Desert Bloom Golf Club, which is just immediately upstream of Medicine Hat, and the other is at the Highway 41 crossing after it flows through the city itself. And again, not a great deal of difference between the two, at least for these compounds that we are measuring. And we have ALS data from, not from these two creeks, but at least on some of these dates that suggest that these are probably not too far off. The effluent itself from the sewage treatment plant, now Medicine Hat is an unusual sewage treatment plant. It's on a trickling filter system. And the trickling filter is based on having a tower that is essentially full of plastic egg crate material. And the liquid effluent is first settled, and what doesn't settle out is then shot into the top of this tower where it trickles down through this entirely biological reactive system that removes organic matter and other things as well. It's then treated by further sedimentation and UV irradiation before it is discharged into a lagoon. And then it spends at least a week, if not more, in the lagoon before it finally makes its way to the river. So even though it's a fairly simple system, it's reasonably effective and it's not overloaded. The wastewater treatment data that we have here are there. As you can see, it seems to be doing its job, at least as far as that. There was nothing there in the first place in the raw stuff, because it's reducing it. And we do have some raw data too. It is definitely doing its job. The river itself, very low concentrations as well. So the effluent itself is not a huge issue for us at this point, especially considering the low volume that's actually getting into the river. And in the river itself, so far as we know at this point, which is based on very limited number of samples, but it looks like it's not a huge issue there either. So to conclude, these things are definitely out there. They're probably out there in greater concentrations than we realize because our sampling program has been very limited. We would like to do more work this summer, particularly with irrigation return water and from the river and from the tributaries, which are more likely to be showing an effect than the main stem river itself. There are very few guidelines. We can adopt some guidelines on a tentative basis, perhaps from other countries, but there are very few Canadian, especially Albertan guidelines for these compounds. But luckily, the concentrations, so far at least, appear to be quite low. The ELISA-based technology that we were testing is something that any reasonably well-equipped lab could do, and that's a big advantage of them. The only piece of equipment you need other than routine glassware and pipettes is a card reader, and these things are relatively common in laboratories for reading 96-well teeter plates for a variety of biochemical tests. So the cost, to give you some idea, you could spend $3,000 for one sample and get a whole suite of analyses by GCMS, and that would be good, except very likely most of them would come back non-detects. Or you could take this approach and analyze representative compounds, and we could probably, with reasonable volume, we could probably do the suite of 10 or a dozen of these things for maybe $100. So you're not going to get the 50 or 60 compounds that the GCMS is measuring, but you do get reasonably reliable data from very representative compounds that could easily give you the information you need. If something obviously seemed to be happening, you could go back in and do more detailed analysis for that. So treat it as an indicator and treat it as a potential monitoring tool. We think that this thing has legs for maybe other WPACs or anybody that's interested in finding out on a short-term basis at reasonably low cost whether they have any major issues in their effluent or in their drinking water or anywhere else. Still, lots of work to be done. We hope to get back out there this summer. Students are coming back, they're keen, and we'll be out there evaluating more kits and doing whatever we can based upon whatever we can scrape together. So, that's my talk. Thank you very much.